So I'd like to welcome Dr. Jeff Coller up, and you're from Johns Hopkins, and you're going to talk about mRNA amplifier technology for SYNGAP1, the gene of the moment.
Oh, it's a pleasure to be here today. Thank you for the invitation, it's wonderful to talk about some of the research that we're doing on RNA therapeutics. And I have to say this is a unique time in history where RNA research is really blossoming into the biomedical space. And this is obviously made evident by the COVID vaccines we all know about. And I've been working on RNA, specifically mRNA, for probably 30 years. And this is the first time I can go to a group of people and use the word RNA and they don't think it means Registered Nurses of America. So I'm going to tell you about my research in my lab. I'm a basic scientist, much like Adrian Craner, who developed the ASO technology for pre-mRNA splicing. My lab works on mRNA, so the mature form, and the intersection between how that mRNA is translated and then the degradation of that mRNA. And the research that we do has been led to some of these therapeutic initiatives. And so, of course, we're concerned about haploinsufficiency disorders. This turns out to be an ideal place to leverage the power of mRNA. And we all know what these are. These are diseases. Oh, I don't have a pointer. Okay, well, these are diseases where you have half the amount of protein that's necessary for appropriate function. And what we're trying to do, much like ASO technology, is to go after the mRNA and improve its expression. And so we really have sort of two ways that we go about this. One is to look at therapies that target specific mutations, patient cohorts with specific mutations. The other is to actually go after the wild-type copy of the mRNA and boost its expression in order to get appropriate mRNA translation. And this really has led into the formation of a company that's called Tevard, and I'll talk more about that in a minute. And what we do in my lab and at Tevard is look at mRNA, the mRNA itself, and leverage the various features that we can to improve its expression. And so we've learned about pre-mRNA splicing, but an mRNA is ultimately what makes the protein. And the three places that we've gone after in making platform technologies to improve protein production is to target the codons that are within the mRNA, and these, of course, are the genetic code that makes the protein. We also go after stop codons. These turn out to be responsible for about anywhere between 25% or 30% of all genetic disorders. And the last technology, which is the one I'll talk about at the end, that we're directing towards SYNGAP, is to work on what's called a poly-A tail. And a poly-A tail is absolutely necessary for the translation of an mRNA, and I'll talk about that in a minute. So again, this technology that was developed in my lab ended up being formed into a company called Tevard, and this is a company that I formed in collaboration with Dr. Harvey Lodish at MIT and Daniel Fisher and Warren Lambert. And Daniel and Warren are two fathers whose daughters have Dravet syndrome, and they wanted to create a gene therapy company in order to go after Dravet syndrome. And we got together and went after these haploinsufficiencies using RNA technology. And Tevard is actually Dravet spelled backwards. And the point of this entire company is to try and turn these recalcitrant genetic disorders around. And so that actually name came from Daniel's son, in fact. And Tevard really has used three technologies that were first developed in my lab to treat haploinsufficiencies. And the first was developed, Tevard was in my lab at Case Western Reserve University when I was there up to about 2 years ago. And this is using small molecules that are called tRNAs. And these are transfer RNAs, and I'll talk about that more in a second. And then a new technology where we're going after poly-A tails. And so I'll start by talking about our tRNA approaches. These are, in fact, very useful for certain genetic disorders, and we have two different types of tRNA therapeutics. One is called the suppressor tRNA, And this is an engineered tRNA. So what a tRNA is a tRNA communicates the information between protein identity or amino acid identity and the genetic code. And so it, in fact, translates, forgive the pun, translates the information of the genetic code into a protein. And we've engineered tRNAs to read mutant patient mutations. These are mutations where usually a stop codon has been introduced, and this causes the truncation of the polypeptide and therefore a non-functional polypeptide. And so we can engineer a tRNA to read that nonsense codon and then lead to restoration of the mutant allele. The second approach is what we call an enhancer tRNA, and this is based on research that came out of my lab where we can enhance mRNA stability using tRNAs to change their expression. Now, the advantages of tRNAs is that they're very small. These are they're kind of like an ASO in the sense that there are only a few nucleotides. There's 75 nucleotides on average. And so this allows us to address the root cause of the disease, as well as target genes that are fairly large. And so most AAV therapies can only are limited by about 4.5 kb. And using a tRNA, we can actually target any large gene. And this also is a one-size-fits-all for many genetic disorders, like Dravet, as well as SYNGAP. Because when we develop a tRNA, like a suppressor tRNA, we can utilize that across indications. So for example, if we have a stop codon mutation, there's only 3 stop codons, and so if we have suppressors that recognize those stop codons, it's portable across multiple indications. And because this is an AAV approach, it's a one-time delivery, so hopefully a one-time and done, and allows us to restore the normal expression only in the cell in which we deliver it, because the tRNA is going to work only on the mutant allele. And so our platforms really go after not only haploinsufficiency disorders, which there are many, but also genetic disorders where there's nonsense mutation. And one of the advantages of these platforms is that we can go after genes, as I mentioned, that are too big, but also these disorders that are called Goldilocks genes. So Dravet syndrome is a classic example of a Goldilocks gene where the underexpression of SCN1A, or sorry, not Dravet, but Rett syndrome, MECP2 is a classic example of a Goldilocks gene where underexpression causes Rett syndrome, but overexpression is also a problem for the patients. And so by working on the mutated copy, we only can restore to the normal amount of the protein. And so that allows us to get within that window. All right, so as I mentioned, tRNAs are critical in protein translation, and they have a vast therapeutic potential. So I'm going to talk about the enhancer approach first, because that's actually the most difficult to understand, and I'll try to walk through it very carefully. And it's based on research that was developed, research from my lab, and as I mentioned, my lab is interested in mRNA stability, how fast an mRNA is degraded. And this one slide sums up 30 years of research, which shows that these 3 mRNAs, these are just what are called northern blots, showing how fast an mRNA goes away. And all you really need to understand is that they're going away at 3 different rates. And every mRNA in the body degrades at a different rate. And that rate of degradation impacts the overall amount of protein that can be made. And so if we can alter those rates, then we can alter the amount of protein that's made. And that's really shown here, where the effect of mRNA decay can be modeled in this fancy little equation up there. The effect of mRNA decay on protein or mRNA abundance is a function not only of transcription, but decay rate. And those have equal effects on the overall amount of protein within the cell. And so what my lab discovered back in 2015 is that the primary determinant of how fast these mRNAs are degraded is in fact the genetic code itself. That each of the 61 words that encode polypeptides not only provides information about amino acid identity, each of those codons is dictating how fast the genetic code is read. And the reason for this is based on the ribosome. The ribosome is the machinery that translates mRNA and makes polypeptides. And this ribosome, which is this blue ball here, or purple, whatever that is, and what it does is every time it sees a codon, it has to find within a cloud of tRNAs around it the right tRNA. And there's about 40 to 50 tRNAs, and this is a random process, so it's constantly sampling the codons. And only the right one wins. And the concentration, how much of that tRNA that exists, dictates how fast the ribosome will move. It's just like if you had 100 ping pong balls and 99 were blue and one was red, it'd be a lot harder to find that red one than the blue one. And so the ribosome's speed is dictated by tRNA concentration. And it turns out there's a machinery that senses how fast this ribosome moves through an mRNA. And if the ribosome moves quickly, then that mRNA is stable. If the ribosome hesitates, though, the machinery that degrades mRNA recognizes that stalled ribosome and degrades it. And this is a normal function of how all mRNAs are regulated. And so again, tRNA concentration dictates how fast the ribosome will move. tRNAs that are high in concentration will stabilize an mRNA, and then tRNAs that are low in concentration will destabilize that. And we can quantitate tRNAs. We can do this using various metrics. And what we had observed back in 2015 is that if we quantitated tRNAs and then overlaid that with how each of the 61 codons impacts mRNA stability, it was in fact a perfect match, such that the 1/3 of the codons that are read fast and are stabilizing mRNAs, and 2/3 are destabilizing. And in fact, this discovery here was used by the lead author, who is Vlad Presniak, to design Moderna's COVID vaccine. And so he is the lead designer of COVID vaccine, made a lot of money. Of course, he didn't give me any of that, but he got a nice sell paper too. So when we made this discovery, it was basically, I'll just show you what you can do with this, is that if you look at an mRNA and recode it with different concentrations of, or different codons that have different concentrations of tRNA, you can modulate its stability. And all you need to see here is that as we do this experiment, we gradually increase the mRNA in a very precise and elegant fashion. And what this means is that we can't do this in the human body. We can't change the codons, but we can change the tRNAs. And so if we could change the concentration of the tRNAs within a cell, we could also modulate stability. So in fact, that's what we do. And so our first platform at Tavard is what we call an enhancer tRNA platform, where we take genes like SCN1A and target very rare codons that have tRNAs that are really low in concentration. And then we overexpress them, and that causes the mRNA to be stabilized. Okay, and so in fact, this works. You can't see it because it's hidden by Tim here, but is there a way to move that? I'm just gonna, yeah, there we go. Sorry, Tim. Okay, and so this is exactly what we're doing here, where in SCN1A, there's 5 regions or 5 clusters of codons within that gene that are that have tRNAs that are of really low concentration. And we can make a cocktail of those tRNAs, introduce them by AAV, and what you can see over here in this graph to the right is that that causes a 1.5 to 2-fold increase in the amount of SCN1A expression. And the beauty of this approach is that it's mutation agnostic. As has been mentioned, diseases like Dravet are caused by any one of 1,800 unique mutations, and so having disease and agnostic approaches, or mutation agnostic approaches, is really powerful for being able to treat the entire cohort of patients. Now the second platform is a suppressor tRNA, and this actually will work for any patient that has a stop codon mutation. And stop codons, of course, the genetic code has within it. You have to be able to start the formation of a protein and then encode it, and then you have to stop that protein from being made. And so 3 of the codons, UAA, UAG, and UGA, are stop codons, and this causes the truncation of the polypeptide. And this often is the reason why patients have patient mutations that cause disease. And the reason for that is really based on the how these stop codons form, which is caused by spontaneous deamination of a cytidine, typically, to a uracil. And so what we do is we play a game where we engineer tRNAs, so there's no tRNA that recognizes a stop codon naturally in the human body. And so what we do is we engineer tRNAs so that they can read that UAA, UAG, and UGA. And therefore what happens is when they experience that stop codon, when the ribosome experiences that stop codon, it now reads through it and forms the normal polypeptide. And it's one of the unique features of biology is that if you look at all mutations that are seen in ClinVar, for example, most patient mutations, up to about 24%, are caused by an arginine, CGA, mutating to UGA. And the other TGA makes up the next 19%. And so you only need a handful of unique tRNA suppressors in order to cover a large population of patients. And in fact, you can, with about 5 of these tRNAs, you can cover almost 95%. And one of the questions we always get with this technology is why doesn't the suppressor read through the normal stop codon? And in fact, it won't. And the reason why it won't is because there's a mechanism to ensure that a tRNA never recognizes the normal termination codon. The normal termination codon has a context around it that makes it impossible for a tRNA to actually read it. And this is something my lab is actually researching, but we think it has to do with the way in which the ribosome experiences that normal termination codon. And we could show you data that, in fact, that you'll never see a tRNA suppressor reading through the normal termination codon. It only goes to the premature termination codon. Here's an example of how this works. So this is SCN1A, a Western blot showing the protein for SCN1A. And you can see in the second lane there's the wild type SCN1A. And this is a cell line that has an arginine mutation at position 1407 to a stop codon. And with no suppressor, you get no expression of SCN1A. But when you add a tRNA suppressor that now incorporates arginine back at that premature termination codon, you restore expression of the protein. Not only do you restore expression of the protein, you restore the function of the protein, which can be seen over here on the right with a patch clamp where you're looking at the opening and closing of the sodium channel. And so it restores it to normal function. In addition, we can use the same exact suppressor to go after a number of mutations. So this isn't SCN1A, this is SCN2A in this case. But in each of these, we're using the same suppressor, or different suppressors as well, and showing that it restores all of the function back to normal for these various alleles. Now, one of the other nice aspects of a tRNA suppressor is that we can take the same drug and repurpose it for a new indication. And so this TR0115 is the tRNA that I was showing you it being used in Dravet syndrome. But here, this is MECP2 levels in a mouse, where we've injected the tRNA into the brain, and with the exact same drug, we can get restoration of MECP2 levels at high dose, shown here in the right in the red coloration. And so tRNAs provide a very valuable tool in our arsenal to go after haploinsufficiencies, but we wanted to continue to play with the mRNA space and how can we continue to improve various haploinsufficiencies. And this leads me to our work on poly A tails. And so every mRNA, this is the structure of an mRNA, they all have a cap at their 5' end. They have a 5' and 3' untranslated region, and then the coding region in between. But then almost every single mRNA in the human body, with the exception of one class, have what's called a poly A tail. And this is added after the synthesis of the mRNA, and the function of that poly A tail, which is usually about 200 to 300 A's in size, is to stimulate the mRNA's translation. And the way in which it does that is the poly A tail binds to a protein that's called poly A binding protein. It's a very clever name. And that function of that protein is to stimulate the translation of that mRNA. And what the function of the poly A tail is that as soon as the mRNA goes into the cytoplasm, that tail begins to be chewed away by deadenylases, enzymes that remove the poly A tail. And so the tail then, as it's being chewed away, it's liberating this poly A binding protein. And so there's one molecule of poly A binding protein for every 25 A's. And so a typical human mRNA has about maybe 6 to 10 copies of poly A binding protein on it. And so what the poly A tail does is it's a slow burning fuse that times how long that mRNA is going to be expressed. And that's very important for appropriate gene regulation, because if you sent an mRNA out into the cytoplasm and it just kept translating and translating and translating, well then that is dysregulation of gene expression. So every mRNA is programmed to basically stop and be destroyed. And that is all through this poly A binding protein. And so what we've been developing and what we're leveraging against SYNGAP is to mimic a poly A tail on an mRNA in order to drive its translation. And shown over here on the right, what you can see is that the longer a poly A tail, the more protein you get on an mRNA. And so that's really what we're trying to do, is augment the expression of the wild-type copy of the mRNA by mimicking a poly A tail. And so the way we've gone after this is really based on a property of RNA binding proteins. And that is that RNA binding proteins are often bipartite in their function. They have to bind an RNA and they have to do something. And those two functions are often separable. And in fact, this was research I did when I was a graduate student in the 90s. Where I had shown that this protein, poly A binding protein, could be separated. You could separate out its ability to bind the poly A tail, but still retain its functional activity in stimulating translation. And so what we do is we insert a artificial tethering activity onto the functional activity of the protein. And that tethering activity allows us to direct it to specific mRNAs. And we do this using a unique protein called DEDCAS13. And DEDCAS13 is an RNA binding protein that uses small guide sequences and guide RNAs. These guide RNAs can be designed to a specific message. And by doing that, we can tether this poly A binding protein fusion to a gene of interest. And as long as that amplifier, that tethered PAB, is bound to an mRNA, that mRNA will translate. And so this was our first experiment that we did with this in the lab, where we just designed reporters, luciferases in this case, and had guides to different regions within the 3' UTR, untranslated region. And what you can see is you can get a nice twofold stimulation of a reporter using this amplifier approach. Now, we don't care about reporters. We care about endogenous proteins, endogenous genes. And so this was a case where we took the same fusion protein, but changed the guide now to MECP2. And MECP2, of course, is responsible for Rett syndrome. And you can see that we get a robust stimulation in those, is that the fourth lane there, fourth and fifth lane, with two different guides that are directed against that mRNA. I'll talk about these other lanes in just a second. The effect is specific to having not only, you have to have the DEDCAS13 PAB fusion. If you just have DEDCAS13, it doesn't work. And so you can see that if you have to have the Cas13 PAB fusion and the guide RNA in order to get the stimulation, and that it works in multiple cell types. And so in every cell type we've ever tested it functions. Now, one of the problems of this approach is that the amplifier itself is way too big to get into a AAV vector. And remember, there's a theoretical limit of 4.7 kb in order to get into an AAV vector. But as I mentioned, these RNA binding proteins are bipartite. We can separate out all these other bits of it in order to and retain its functional activity. And so this is a structure, in fact, over on the right, of poly-A binding protein in white and salmon color, fused to dead Cas13. And the white part is the part that binds poly-A. And so we don't need that anymore because we have it fused to dead Cas13. And the salmon-colored part is the part that stimulates translation. And so what we're doing is we're just getting rid of all that stuff in between, and we wanted to make sure that this minimal mRNA amplifier still folded correctly as a protein, and that's what this is showing here, is that when we make a mini mRNA amplifier where we just have this domain is actually called the Mademoiselle domain. When you have the Mademoiselle domain alone, it still folds in the correct way, and so does dead Cas13. And the last 3 lanes in this Western that I already showed you, you can see that that minimal poly-A binding protein still functions, and it functions almost to the same extent as the full-length form. And so this is a minimal form that actually could easily fit into an AAV vector with a guide sequence. And it's, in fact, these molecules that we're continuing to develop. One of the beauties of this is that 3' untranslated regions are large in humans. They tend to be like 1,000 nucleotides in size. And so we're not limited to a specific area within a 3' UTR to design the guide. And what we've seen is that where we place the guide within the 3' UTR modulates the amount of protein that we get out. And you can see that in this experiment here, where as we move the guide closer and closer to the 3' end in this case, we get more and more expression of MECP2. And so we think there's the ability to modulate how much protein we're getting out by changing guides and their location. And as I mentioned, it works in many different cell types. This is just an example of that. Okay, so this really is a mutation-agnostic technology then, again, that raises the expression of the wild-type copy of the mRNA. And it's very portable across indications because we can simply change the guide, and I'm showing that here. And so in panel A, we've designed a guide against SYNGAP1, and you can see we get a nice 2-fold stimulation of that mRNA with 2 different guides, as well as these other genes that are associated with haploinsufficiencies such as SHANK3, PTEN, and CHD2. And so we're happily funded now by the SYNGAP Research Fund to continue to develop this technology on SYNGAP and show efficacy in iPS cells and hopefully in mouse models. All right, so the mRNA amplifier approach is, again, a way to tether a mRNA translational activator to an specific mRNA and enhance its translation. And we're excited to continue developing that. And so we have at least 3 different platform technologies that we're developing at Tevard that have come out of our lab and can be used on multiple indications. And we'll be telling you much more about these studies hopefully in the very near future. And so this is Tevard's leadership. This includes Harvey Lodish, who is one of our co-founders and SAB chair. And we're pleased to have some of the world leaders in tRNA and gene therapy space on our leadership board. And with that, I'll just thank the people in my lab who've done all this work, as well as the people on the right who give us money to do these sorts of things, including you guys. So thank you very much.
Wow, that's super, super cool. Can we have a microphone over here, please? And while we're doing that, I just want to ask a question. How often do you have to give these drugs?
Well, AAV would be one and done, hopefully.
That's what I was hoping you'd say. Please.
Hi, have you looked at combining the enhancer and the poly-A approaches?
We think that we could. And that actually is a really important point, is that many of these technologies, especially the tRNAs, are small, very small, and the amplifier, it's not so small, but you could easily put the tRNAs into the same AAV vector. And so there's the possibility that you could, if a patient had a stop codon mutation, for example, and you only get, let's say, 10 to 20% efficacy with that stop codon suppressor, if you combine that with an amplifier, you might get over that therapeutic mark, into a therapeutic window that provided efficacy. So. Yeah, Victor.
Yeah, at the back
Oh.
over there. Oh, so you saw someone.
I saw back there.
Yeah, we saw it. Oh, no, there you go.
Oh, oh,
Who's got the mic?
oh.
Over here.
She's
Over
right there.
here.
Okay, I thought it was over there.
No, take the mic because Tim's online too.
Okay, sorry. So I realize that you've already addressed this generally in talking about being able to modulate the amount of protein and specifically in Rett syndrome, like being able to hit a specific range. I'm just curious, from my personal experience, when my daughter was diagnosed 2 years ago, our geneticist told me directly, exactly, I wouldn't hold my breath for a treatment on this because it's not a good candidate, based on the fact that you can also have too much SYNGAP, causing things like schizophrenia and other also awful things. So anyway, I guess I'm just curious to hear your thoughts on that. I mean, I assume obviously you disagree. Do you think that's...
I do disagree. What did Ed say about idiopathic?
We won't go.
We won't go there. I do disagree because I think, one, there's a number of... What's happening now in medicine is really unique because we have the ability to sequence patients and identify the mutation that they have. And we do that readily, and that goes into a diagnosis and a treatment plan. And, our technology is what it is, but you also just heard about, what Stoke is doing and what other people are doing with ASOs. All of these, we want all of these to succeed because some patients will benefit from one technology, some patients will benefit from another technology. I do think we can tailor some of our approaches to get within a therapeutic window. It's amazing what we can do now with gene therapy. I mean, it's really a revolutionary time for rare diseases. So I wouldn't, say that there's no hope for that just because of the possibility of overexpression. I would say we've got to figure it out how to make sure it doesn't happen, and we will.